it's my privilege to introduce Robert Anthony. So, if you Google David Rollins, you'll find a guitar player. If you Google Robert Anthony, you'll get a WWE wrestler, but this is not a WWE wrestler. He's an old friend that's done nothing but amazing work from his postdoc or PhD all the way up to, his faculty position here, publishing in Cell Science, Nature, all through his stage careers. But I really appreciate how he's been a Renaissance man in terms of he always has a good book to read. He always has a good band recommendation. He has his own band CD that you can actually go on and buy and purchase. But he's not here to talk about those things. When he was a PhD candidate, he worked with Bill Gauss and discovered kind of mechanisms of Th2 memory. So identifying that CD4 T cells can really instruct a rapid alternative activated macrophage. He went on to join Jeff Ravich's lab at the Rockefeller doing just stellar work on sialylation as a mechanism for IVIG. So IVIG is something we use here in primary immune deficiencies for immunoglobulin replacement, but in rheumatology and other fields, we use it for the immune suppressive qualities. And that was not an appreciated mechanism. We really didn't know how it worked. And so the sialylation, I think, was a really great discovery. He's gone on to start his lab on glycosylation and really novel thinking about how you can do glycoengineering in vivo. And I'm going to introduce this talk, non-templated regulation of antibody function, and hopefully I can follow.
Eric, thank you for that very kind introduction. Thanks to the organizers for the invitation to be here. It's really nice. I've had a lovely time in Seattle. I've got 2 small kids at home, so to be able to just have a couple days and not have to be a dad full time is always a treat. Like the night in a hotel is lovely. So I've known Eric for almost 20 years. So if anyone wants some dirt, we could certainly talk after. But yeah, so we spent a lot of time hanging out when Eric was at Penn as an MD PhD student when I was in graduate school. And so I'm happy to keep this talk as informal as we'd like. So if there's any questions that you want to ask, as they arise, please feel free to do so. And so here is a schematic of the human heavy chain locus on chromosome 14 with schematics of all the antibodies that these heavy chain produce. And I've kind of put approximately where there are in-link glycosylation sites on all of the antibodies in the constant regions of them. And it is very well established that glycosylation is important for IgG1, but it's not clear what glycosylation does to any of the other antibodies. So this really is a wide open field, and something that I think I started to wonder what it did to the other antibodies when I started my lab about 10 years ago. So this is a molecule I think everyone here is familiar with. This is actually the B12 HIV neutralizing antibody, but the top of this molecule is responsible for the specificity. And there's a lot of people in this room that are absolute experts in this part of the molecule, and I'm really just an idiot savant. I don't know anything about the Fab at all. So I'm very interested in the constant region of the antibody, which is the business end. And so there is a single N-link glycan, asparagine 297, that's conserved across all mammalian IgG antibodies. And so that's an indication of how important this glycan is for antibody biology. And the canonical functions of antibodies are triggering inflammation, right? So that's in the form of antibody-dependent cytotoxicity, opsonizing antigens, and they can also fix complement that may occur in some organs at the end stage of autoimmune diseases. And so this is just a picture of my postdoctoral mentor, Jeff Ravich. And so Jeff went on to show how antibodies work primarily. And so this experiment that was done, many years ago by Raphael Kleins, A monoclonal antibody that's specific for the platelet integrin was administered to mice, and within hours, there's rapid depletion of platelets from the circulation, which is what you can see in this red line. Mice that were given an isotype control antibody in green have no loss of platelets, and then the C3 knockout mice had a rapid loss of platelets mirroring the wild type treated animals. And C3 is the complement protein that's essentially common to all three cascades. So this really showed that complement was not essentially mediating the depletion of platelets. There we go, right. And so the signaling component of activating Fc receptors, the gamma chain, when that was depleted from these animals, there's no loss of platelets. Essentially, in this mouse, phenocopies the isotype control animal. And so this experiment showed that to deplete platelets by an antibody, you needed activating Fc receptors. There, okay. Sorry, there we go. And so to interact with an Fc receptor, that glycan is crucial. So these are essentially human wild type and then a mutant antibody where the glycosylation site is disrupted. And you can see this is confirmed by lectin blot here. And these are just affinity constants from Viacor experiments looking at the interactions of a monomeric antibody with the different Fc receptor polymorphisms that are found in the human population. So you can see that a wild type antibody has essentially moderate to low affinity in the micromolar range. So essentially forming an immune complex, you'd increase the avidity and get a productive interaction of the Fc receptors, but that's totally ablated when you basically remove that single end-link glycan. All right. I think we should have just, dragged it onto the hard drive. Anywho, so this is just a schematic of the end-link glycosylation pathway. And so in the ER, there's this 14 sugar precursor that's added to proteins, and then as they pass through the Golgi, the glycan structure is trimmed down, and then it could be built back up. And ultimately, there are three main types of end-link glycans: these oligomannose, hybrid, and the complex binary structures. And it's not totally clear how this is regulated, but there's some things like the steric accessibility of the glycans to various glycosidases or glycosyltransferases, and the availability of the sugar nucleotide donors may play a role in influence in this. But IgG antibodies have these complex binary structures predominantly on the Fc. So this is just a schematic of the complex binary glycan on an IgG Fc. And what I think is pretty interesting about it is that this is remarkably heterogeneous. So there's over 30 distinct variations of this in the serum of healthy individuals on antibody Fcs. So all of the antibodies have this core structure within the dotted box, but they vary by the presence of fucose, bisecting glucnac, or the arms can be extended by galactose or sialic acid. And the reason that I think this is important is because the various composition of sugars that are present exert profound influence over the function of antibodies. So for example, antibodies that lack fucose gain a 50-fold increase in affinity to Fc gamma R3A. And this is the Fc receptor that mediates antibody-dependent cytotoxicity. So basically, afucosylated antibodies are much better killers in vivo than fucosylated antibodies. And so a lot of next-generation therapeutics are being developed to lack fucose so they can be more potent in vivo. And then terminal sialylation, which caps the glycan, that actually impairs binding to Fc receptors. So antibodies that have sialic acid are not very good at killing or depleting cells, but they're also thought to be essentially anti-inflammatory and the biologically active component in high-dose intravenous immunoglobulin. All right, and so this is just a MALDI-TOF of IVIG, which is essentially representative of a normal, healthy individual's Fc glycans. And so you can see how many different glycoforms there are that are present. And approximately 10% of these glycans terminate in sialic acid, and these are just represented underneath the red brackets. But the vast majority either terminate in 2-galactose, 1-galactose, or no galactose. So I talked to a couple of people about intravenous immunoglobulin today. So I know that there certainly are some experts in the audience. But for those of you that are not, it's a therapeutic preparation of monomeric IgG antibodies from tens of thousands of donors. It's developed as an antibody replacement therapy, and it's given in that capacity at 400 to 600 mg per kg to immunocompromised individuals. In the 80s, out of desperation, a Swiss clinician gave essentially a 5-fold higher dose of the same preparation to pediatric ITP patients. So this is an autoimmune disease where platelets are attacked by autoantibodies. And that higher dose restored the platelets to normal levels. And it's been approved in that capacity for a handful of indications by the FDA and other regulatory agents. But it's given an absolute ton off-label in this capacity. So it's the same preparation that just has completely paradoxical properties at different doses. And so when I was a postdoc, we were trying to figure out how this could be. And so one of the models that we use is this KBXN serum transfer model. And this was developed by Diane Mathis and Christophe Benoit when they were in Marseille. And they essentially made this KRN TCR transgenic line and crossed it to a NOD line. And that's where the nomenclature of this progeny comes out. It's this KBXN strain. They developed spontaneous inflammation of the paws that's caused by autoantibodies. And you can transfer that to almost any recipient strain with some very notable exceptions. But when you do the transfer, you get this characteristic edema of the paws and a very characteristic inflammatory infiltration that's neutrophils and macrophages to the joints. And then importantly, from our perspective, the immunomodulatory dose of IVIG, so 1 gram per kilogram, prevents the swelling and it prevents the inflammatory infiltration. So it's just a clear readout in vivo of anti-inflammatory activity of IVIG. So right before I joined Jeff's lab, they did a very simple experiment where they just removed the glycans from IVIG with the enzyme PNGSF. And that was just confirmed by this lectin blot. And then they tested it in the KBXN model. And you can see by clinical score, the serum transfer was fairly robust at triggering inflammation, shown in gray. IVIG prevented the swelling, the clinical score about 2 in red. And the deglycosylated preparation lost its activity. So this suggested that the glycan, while it's also known to be important for binding to Fc receptors and the pro-inflammatory activity, it's also important for the immunomodulatory activity of IgG. And then they removed sialic acid. Removing it's pretty easy to do. You can just get a neuraminidase from like Leland Biolabs. And so the digest was confirmed here, and the results are shown in this graph, where you can see the sialylated IVIG protects, but when you remove sialic acid, you lose the activity in this in vivo model. And so when I was a postdoc, we essentially wanted to make a recombinant preparation of hypersialylated IVIG. So we generated recombinant antibodies, made FCs and separated them, and then we put on galactose and sialic acid in a stepwise reaction. It's just shown here. And we showed that essentially a 30-fold lower dose of these hypersialylated FCs was able to protect in the model similarly to the high-dose IVIG. The 30-fold lower dose is just an equal molar concentration of the sialylated antibody. So essentially, if 10% of antibodies in IVIG have sialic acid and the FC is a third of the weight, we did the hard math and got 30-fold less. So, and this general approach was being developed by Momenta Pharmaceuticals, where they were doing literally that stepwise preparation to make hypersialylated IVIG. And then they had a press release a couple of years ago how it seemed to be working in ITP. And then Momenta was bought by Janssen, and Janssen's trying to license, I think, this to Takeda, but I've lost track of where it is. But this we interpret as vindication of our studies and also using the KBXN model as a good way to understand how IVIG works mechanistically. And so some of the work we also found was that, as I mentioned, sialylation impairs what Momenta is developing is just they're taking IVIG off the shelf and hypersialylating it. What we made was just the FC itself. So there's no Fab at all. But so I mentioned earlier that sialylation impairs binding to FC receptors. So here we're using the inhibitory receptor, FC gamma 2B is just a model FC receptor, but it's true for all the low affinity for FC receptors. But you can see in this B core trace, you have this impaired binding of a sialylated antibody, while there's this nice dose-dependent binding for an antibody that lacks sialic acid. And so what What sialylation does is it completely converts the receptor binding altogether. So you can see binding to the C-type lectin DC sign, which does not interact at all with an antibody lacking sialic acid, shown here. All of a sudden, we get this nice dose-dependent binding in a totally different curved shape with the molecule DC sign. And so DC sign is essentially a member of the sign family of receptors. There's 8 different sign receptors in the mouse and 2 in the human. And these are polysaccharide recognition receptors for pathogens. And SINR1, it has the highest homology in the carbohydrate recognition domain to human DC sign, but the expression is slightly different. And so, so we made a back transgenic mouse, essentially inserting the human DC sign locus into a mouse and then back cross that onto the SINR1 knockout background, and tested that the anti-inflammatory activity of hypersialylated FCs in this animal. So you can see in a wild-type control, the sialylated FCs were anti-inflammatory. On the SINR1 knockout background, they lost the activity, which is shown in these red hatched bars. But when we put the human DC sign back on that background, you could actually restore the activity. So this really showed that in vivo, DC sign or SINR1, depending on if it's the mouse or the human system, is really conveying the anti-inflammatory signal of the sialylated antibodies. And so when I started, when I moved up to Boston, we knew that sialylation was really important for IgG1. But we wondered about what are the other 4 classes, or 3 additional classes of IgG antibodies. And so I think everyone here knows, but there's 4 different types of IgG in humans, numbered 1 to 4. Numbers IgG1 and IgG3 are fairly potent, so they can both fix complement and bind to Fc receptors, so they're pretty good at killing target cells in vivo. But IgG2 and 4 are fairly poor binders of both complement and Fc receptors. They're thought to be either inert or anti-inflammatory or inhibitory, depending upon essentially what school you go to. But the role of the glycan in all of those subclasses of antibodies is not known. And so Ravi in my lab generated recombinant IgG 1, 2, 3, and 4, and then using that stepwise sialylation reaction, put sialic acid on them. So you can see this is just our IVIG loading control, and we're releasing the glycans on the FC and running it over an HBLC column, so it has all of these different peaks. We've just color-coded it by the terminal glycan. So essentially you can see that glycans that terminate in 1 or 2 sialic acid are pink and purple, that terminate in 1 or 2 galactose are essentially yellow or gold, and then if they have no galactose, they're blue. And so you can see that essentially coming out of tissue culture, most of these antibodies have either no galactose or 1 galactose, but it still is reasonably heterogeneous. But then when we put on galactose and sialic acid, you can shift the peak primarily to these monosialylated glycans. And so we tested them in the K-Bixin transfer model, and what you can see is that when we sialate an IgG1 antibody, which is essentially compared to the acylated in blue and acylated in red, it's inhibitory. When we sialate an IgG2 antibody, there's no effect, so it's not anti-inflammatory. When you sialate IgG3, all of a sudden it's anti-inflammatory, and then when you sialate IgG4, there's no protection. So we thought this was pretty interesting because the anti-inflammatory activity that's conveyed by sialylation tracks only on the antibodies that have pro-inflammatory activity. So it really seems to be held with these antibodies that already bind to Fc receptors.
Those all at the same asparagine.
Yeah, so it's all that N297 site, absolutely. And so we made a immortalized bone marrow-derived macrophage line. This was actually done by Kate and Isabella, who were at NJH at the time. And so we did this for the humanized DC sign mice, and then the background control for SINR1 to have a reagent that we could test DC sign interactions with. And we looked to see whether or not the different subclasses of IgG could still bind to DC sign or not. And as you'd probably predict, so this is just the characterization of these lines that was done by Anna. And you can see that really the only difference is that in the humanized DC sign line, there's DC sign positivity that's not present in the SINR1 knockouts or in the wild type controls. And so Anna took essentially recombinant IgG 1, 2, 3, and 4 with or without sialic acid. and put them on these cells and looked for binding. And what you can see is that IgG1 binds to the DC sign cells when it's sialylated, but not when it's lacking sialylation. There's no detectable binding for IgG2. IgG3, when it's sialylated, binds to DC sign, but not IgG4. So the in vivo activity is absolutely consistent with DC sign binding. We turned back in vivo, so looking at a SINE1 deficient animal, you can see sialylated IgG1 does not protect, IVIG does not protect. That's also true for sialylated IgG3, but when we put it on the DC sign transgenic, all of a sudden they're protecting again. So essentially the pathways of anti-inflammatory activity seem to be conserved when we sialate a G3 as well as an IgG1. And this just came off earlier this week. I'm really excited about this. But we aligned the FC sequences of all of the different allotypes of the human IgG antibodies. And we were looking for residues that were conserved in IgG1 and 3, as these are the ones that are anti-inflammatory when you sialate them, that are different in IgG2 and 4. And so we've colored them all in ways that I think we try to make sense. And what you may or may not be able to see is that essentially there are 2 residues that fit this criteria. So essentially there's this residue at 2, 3, 4 that is shared between IgG1 and 3 that's different in 2 and 4, and also at 3, 2, 7 that's essentially that's conserved, that's in IgG1 and 3 that's different in 2 and 4. And so Sunny is in the process of essentially introducing the residues of IgG in 1 into IgG4, and then she's going to sialate and see if that's an anti-inflammatory and doing the reciprocal mutant. So my hunch is that essentially you need to have, is it a lysine? A lysine at this position with sialic acid to actually be anti-inflammatory. But we're going to do all the different iterations to show that. So anyways, hopefully we'll be able to show that definitively in a couple weeks, but this is still very much a project under works. And so, right. So we think that there's something about that leucine residue that there's actually a conformational change of IgG from a closed to an open conformation that would influence whether or not it binds to it, like a SC gamma 2 versus the DC sign. And so I'm hoping that we're going to do some kind of structural modeling, but that leucine actually allows it to be flexible and perhaps IgG2 and 4. We hope, we'll see. But we're going to do all the kind of genetic stuff. But we think it'd be pretty fun. I mean, it's just unreal to look at, essentially how conserved it is. So we had the first iteration of that figure, we only did 1 allotype of all the antibodies, and then we just put all of the allotypes in, and it was totally, it really is conserved across all of the different antibody sequences. We don't know. So there's not a structure for IgG binding to DC sign. There's a structure of IgE binding to CD23. And CD23 is actually on the same chromosome as DC sign. They're right next to each other. So it's quite possible that was a genome duplication. And then if you superimpose the structure of DC sign for CD23 and IgG for IgE, there's remarkable heterogeneity. But that's kind of a lot of assumptions in that model. So yeah. Right. Yes, thank you for reminding me to do that. Yes, so the question is, what happens when DC sign is bound? So DC sign is expressed on macrophages and dendritic cells, and we don't know what's happening inside of the cell. Like, that's one thing we're working on. So Anna's project is to look at the signals and essentially the gene expression profiles that are conveyed after ligation. But some of the work we did in Jeff's lab was we found that there was IL-33 was produced by these macrophages that seem to activate an expansion of basophils to produce IL-4 that would then increase the inhibitory Fc receptor expression on inflammatory macrophages. And with that essentially upregulation, they were less receptive to trigger inflammation by immune complexes. Yeah.
Do we know if this sequence likes it activates inflammatory or an activated pathway, like does it have, ICANS or actions or whatever?
So yes, what's known about DC sign signaling, I would say, is pretty unsatisfactory. So it was discovered a number of years ago by Theo Gietenbeek, and he did some beautiful biochemistry. It binds GP120, the HIV glycoprotein, very strongly. So that's it has a ton of oligomannose residues on it. And but to get it to signal, all of the studies required a secondary signal. So they would put GP120 and LPS on. And so then they were really tracking differences of the toll receptor signaling pathway. And so that's all the signaling work. You need a secondary signal. And I mean, unless our preps are like woefully contaminated, I don't think there's a secondary signal in there, right? So we're trying to see what's going on. It's certainly possible that they're going through an FC receptor, like SIC or SHIP signaling as well. So we're trying to be agnostic to that. But yeah. But yeah, those experiments are kind of in the works, and we're trying to really get the other story about what amino acid is out before we do the deep dive into the signaling. But that's totally the direction we're going to go. So the other antibody class that my lab is interested in is IgE, which is shown over here. And so while there is one glycosylation site on human IgE in the constant domain at asparagine 297, there are 7 glycosylation sites on the heavy chain of human IgE and 9 on mouse IgE. So it's much more heavily glycosylated. So IgE is, I think everyone in here knows, it's responsible for allergic disease. So it binds to the surface of mast cells and basophils that express the high affinity IgE receptor, FcεR1. And atopic diseases are on the rise all over the world. I think 1 in 5 individuals worldwide has some sort of acid. Yeah, so essentially, when IgE is cross-linked on the surface of mast cells, you get degranulation and the release of these mediators that are responsible for the symptoms of allergic disease, like histamine, prostaglandin, and leukotrienes. So when we started this project, we compared essentially the structures in the FC of IgG and IgE. And IgG is shown over here on the right. With that's the CH2 and CH3 domains. This structure was generated by Peter Sonderman. And this is Ted Drzezinski's structure of IgE, really just focusing on the CH3 and CH4 domains. And I'm not a structural biologist, but I found the similarities to be remarkable, not only in terms of the protein, but there is a single N-link glycan on each of these molecules that is present in similar spots, but it's also resolvable by crystallography, right? So the glycan on the IgG molecule essentially lines this stretch of hydrophobic amino acids. And so it has a fixed position in the molecule. You can see right here in this structure, there's a glycan that's stuck right there that Ted Drzezinski was able to resolve that in his work when he published this at the turn of the century. And so this is just an amino acid alignment of all of the mammalian IgE antibodies, including the ductile platypus. And what you can see is there is a glycosylation site that's conserved across all of these, and that's at asparagine 394. And so we asked, what does the glycans on IgE look like? Because there's 7 N-link glycosylation sites. And so Wayne Strothler is a pediatric allergist. His lab is right next to mine. And so he has serum from allergic patients. And so we enriched for IgE, and that essentially subjected it to glycopeptide mass spectrometry. So to do this, we would take the purified IgE and then digest it with trypsin and chymotrypsin to generate glycopeptides that we then run over the mass spec. And this can give us essentially a site-specific picture of what each glycan is across the molecule. And so the results are shown here. So this is total IgE from an allergic patient. And so you can see that the conserved site in the 394 has these oligomannose glycans exclusively. And so these are formed very early in the N-link glycosylation pathway, which is shown, yes, so the structure is shown here. Essentially this N383 site is not occupied. N371 is partially occupied, but all of the sites except for N394 have a ton of sialic acid. So they're either mono or disialylated glycans that have the complex binary structure. And so we could interrogate what the conserved glycan was doing using different glycosidases. So PNGSF will cleave all in-linked glycans. NOF1 will cleave only oligomannose glycans, and it cleaves between these two different GLUCNAC residues. And so we treated IgE with either buffer or NOF1, and then we gave it systemically to mice, which is shown here. And then the next day we would just gavage, and this is a model of what we're calling passive food anaphylaxis. And then you can measure body temperature over about an hour or so every 10 minutes. And so if you get an anaphylactic response, you get a drop in temperature upon giving the allergen. So we did this experiment, and you can see we get about a 2-degree drop in temperature in buffer control treated IgE, but when you remove that single oligomannose residue with an enzyme NOF1, there's essentially no drop in temperature. So we took IgE with this time it's human IgE that we treated with either buffer or NOF1 and loaded it onto the surface of these LED2 human mast cells. And then you can cross-link the mast cells and then measure for the release of beta-hexaminidase in the supernatant as a surrogate readout for the magnitude of degranulation. And so when we did that for a recombinant OVA-specific human IgE, you can see that essentially the recombinant IgE shown in black was pretty robust at triggering anaphylaxis. When we removed the glycan, shown in these solid pink bars, there's absolutely no degranulation, but the buffer control in the open bars acted fairly similarly to the wild-type treated IgE. And that's true also for primary IgE that was cross-linked with just anti-human light chain, so an antigen agnostic way. So essentially the conserved glycan is essential for triggering mast cell degranulation. When we remove that glycan, it totally kills binding to the high-affinity receptors. So you can see these Biacore traces of the buffer treated. This off rate is 3 days. So it's an incredibly strong interaction. And just removing that single glycan, there's no detectable interaction with the high-affinity receptor, which I think is pretty remarkable. So we tried to gain some insight into the secondary structure by using circular dichroism. So this looks at essentially absorption of left and right polarized light over a number of different wavelengths. And so you can see the CD traces here. And so looking at a wild-type IgE in black or a buffer control treatment in green, they overlap very well over these different wavelengths. But when we remove that single glycan shown in pink, there's a slight shift, which is consistent with the conformational change upon removing a single glycan. So we think that's essentially the mechanism of how that oligomannose is, when it's removed, you ablate binding the high-affinity receptor. But we have a collaboration going on. We're trying to actually get some structures with a deglycosylated IgE antibody. And so Katrin Sitting, who is a German master student in the lab, she wanted to know what part of this oligomannose was required for binding the high-affinity receptor. So she devised this scheme on how to systematically remove different residues from the structure. And that's just outlined here. And so the first experiment she did was to treat IgE with a 1,2,3 mannose. And so that would generate essentially this MAN3 structure. And I think it's just worth reminding that mannose is only going to work on the oligomannose glycans because the complex binary glycans do not terminate mannose. And so she confirmed the digest first by HPLC, and you can see that while IgE, the oligomannose exists with these 5, 6, 7, 8, 9 mannose structures, they all shift to a MAN3 structure by HPLC, and that's just quantified here. And then she did some of the biochemistry where you can see the lectin blots and the Kamansi. And then she tested it in functional models. So first she did the passive cutaneous anaphylaxis model. So she injected IgE, either the wild type of the MAN3 IgE into the ears of mice intradermally. And then the next day she challenged with the allergen in Evans blue dye. And if there's a productive anaphylactic response, you'll get essentially a blue coloration of the ear that you can quantify as a surrogate readout for the magnitude of anaphylaxis. Right, and so this was the experiment. You can actually see representative mouse ears here, but comparing the wild-type IgE with the MAN3 IgE, you can see there's no difference. and then in blue is the Indo-F1 treated IgE, which lost all the activity. So this MAN3 structure is still totally functional for an IgE. Looking at degranulation of human IgE on the mast cells, you can see comparing the wild type structure with the MAN3 or the buffer control. It still triggers pretty robust degranulation, especially compared to the Indo-F1 treatment. So again, it's functional for human IgE as well. And then she wanted to make sure it was intact. And so this is just looking at binding to the allergen, which still it does a nice S-shaped curve for both the IgE and the MAN3, the buffer control. So, and then using an octet assay to bind to the high affinity receptor, you can see that both the wild type are untreated and then the MAN3 bind fairly strongly. So these antibodies still work, can bind to the high affinity receptor, can trigger degranulation. So then she took IgE and treated with a 1,3,6 monocytase, generating this MAN1 structure. That was quantified here by HPLC. You can see it's barely coming off in time for us to capture it, just right there. All right, so in the cutaneous anaphylaxis model, you can see that this MAN1 structure is still triggering a fairly robust anaphylactic response compared to the untreated control and also compared to the Indo-F1 control. So it seems to be working in the mouse system. In the human system, this MAN1 glycan shown in green here is still triggering robust degranulation of the human mast cells, especially compared to the Indo-F1 control. And then it still binds to allergen and still binds to the high affinity receptor. So again, the MAN1 structure is still a functioning IgE. And this was fairly comforting because the IgE that was crystallized by Ted Drzezinski was generated in insect cells, which had a MAN1 structure. So this is all consistent with what we would have guessed would have happened. And so Catherine then took the MAN1 IgE and treated it with a beta monocytase to generate this MAN0 structure. And just because the MAN1 structure was barely coming off the HPLC, we turned to mass spec, and Brian Maurice ran this over our Q-Exactive. And this is what he found, was that essentially there was predominantly these MAN1 structures on the alpha-1, 2, 3, 6 monocytase treated IgE, but when we added the beta monocytase, getting about 80% that terminated in a single N-acidic glucosidase. So that was nice. And then at that point, Catherine had finished her master's thesis, had to go back to Germany. So Shayatan was, he's picked up this project, but you can see we're starting to see about a 50-fold reduction in degranulation as shown here in the human mast cells when we clip off that single last mannose residue. And so we really think that essentially when you just get rid of all the mannose, that's when this interaction starts to be impacted.
Do all those isoforms exist? Like if you just take polyclonal IgE, you talked about all the different forms of IgG, but do all the different isoforms or kind of different glycosylations exist naturally in our IgE?
Right. So the question is, what does the glycans in IgE look like naturally? So at the oligomannose structure for allergic individuals or non-allergic individuals, the oligomannose exists as either a MAN5, MAN6, MAN7, 8, or 9 structure. So it's a fairly processed structure. We're starting now to look at some other disorders that have either hyper IgE or systemic or lupus, where lupus nephritis and Helminth infected IgE, and there are some strange things in hyper IgE syndrome. So that's the only condition I've seen where it's not a fully processed MAN5 structure. So these are essentially, this is just the biochemistry of this what we're calling the MAN0 IgE structure. But looking at antigen binding, you can see it still binds similarly to the buffer control, but we're really seeing quite a reduction in affinity when you have the MAN0 compared to the MAN1 IgE. And I think it's interesting to remember this is still probably able to get some productive engagement with the high affinity receptor, but not nearly as robust as what you would see if there's a single mannose there present. So another question my lab is very interested in is this idea of what makes IgE pathogenic or not. And it's really, this is a fairly old report, but But what it illustrates is that all of these patients were presumed to be allergic to a particular food. I think everyone here knows, to diagnose food allergy, somebody would be experiencing fairly general symptoms. They'd be vomiting or have hives, have itchy type reaction, and then there'll be a case history that's taken. And if there's some allergy associated food, the person would start avoiding it. And so essentially, these patients were presumed to be avoiding all of these different foods. In a clinical setting, they had an oral food challenge, and it was found a fraction of these patients actually reacted to the food they were presumed to be allergic to. And so in the case of peanuts, there's about a 50% false positive rate. And that means that 50% of the people that presume they are allergic to peanuts, either because they have a skin prick test where peanuts are injected intradermally and they have a classic Whelan-Ferry reaction, half of those patients can ingest peanuts and not have any allergic reaction. So we're really interested in why is that? How can people have peanut reactive IgE that triggers a classic Whelan-Ferry response and not be allergic? And so there's a number of non-mutually exclusive explanations for this. One is there could be allergen-specific IgG that might be present in some patients that might block the interactions with IgE and the allergen. One might be there's lower affinity of IgE for the allergen. That might explain some of these differences. There might be Tregs involved that inhibit the reaction. There could be differences in the signaling threshold of mast cells based on the receptor density of either the high affinity receptor or essentially SYK. But we asked whether or not there are intrinsic differences in the IgE molecule itself. And so in collaboration with Wayne and Sarita, who are neighbors, they had this really great cohort of peanut allergic patients, and these were confirmed by essentially oral peanut challenge. So these are bona fide allergic patients. And then we got a cohort of absolutely non-allergic adults, and did a very simple experiment where we just loaded the total IgE, the same amount, onto the surface of mast cells and cross-linked them with anti-IgE, so an antigen-agnostic way. We got this very interesting trend where there was always slightly less degranulation in the non-allergic IgE compared to the allergic IgE, and that's just shown in blue versus red. And so Michelle asked whether or not there was differences in glycosylation between the two different IgE cohorts. And so first she focused on the N394 glycans, which are the Oligomannose, and you can see just the different general structures. And so this is just kind of getting back to what you were asking about, Eric. You can see that there's basically these mannose 5 through 9 structures that are very similar on the IgE in both different cohorts. So we started looking at the variable glycans that are different on the IgG molecule, because that certainly informs our presence of how antibodies work. So looking at fucose, we saw no difference looking at IgE on non-allergic and allergic. We started to see a slight difference in this bisecting inositol-glucosamine, which was a little bit enriched in the non-allergic IgE. But we got very excited because there was a clear enrichment of terminally galactosylated glycans in the non-allergic IgE, and conversely enriched sialylated glycans on the allergic IgE. And obviously these are related because sialic acid is attached to galactose. And so we did the ROC plots, and you can see that essentially changes in both sialic acid and galactose tracked with the incidence of disease, while changes in the other sugars did not. And so Kai asked whether or not sialic acid did anything to IgE. So she took mouse IgE and removed sialic acid with neuraminidase, and then did the PCA model. And so these were the results that she got, which were pretty exciting, where since you can see the sialylated allergic IgE is robust at triggering an anaphylactic response. You can see the image of the ears right here. Which removed sialic acid, it completely attenuated anaphylaxis. And that's just pictured here and then quantified. And then Kai was particularly paranoid about results. So she put sialic acid back on in vitro, which is this resialylated preparation. That just restored anaphylaxis. And there's just a picture of the ear right there. So this really showed that the presence or absence of sialic acid sets the threshold of anaphylaxis in vivo. And so it's true also for human IgE on the LED2 mast cells. You can see when we loaded them with either sialylated or acylated IgE and then cross-linked with the allergen ova, there's at least a twofold reduction in degranulation when we sensitized with the acylated IgE. So how this is happening, we don't know. But this is binding to the high affinity receptor. You can see there is really no difference with and without sialic acid between the two different molecules. Still a very strong high affinity interaction. Binding to allergen, which is in this case is ova, you can see it's still intact with and then without sialic acid. But the signaling is attenuated. So looking at SIC, which is immediately downstream of the high affinity receptor, 5 minutes after cross-linking, you can see in the acylated preparation, there's less phospho-SIC compared to the sialylated controls. And then looking at calcium flux, which is downstream of all of the signaling, you can see there's reduced calcium flux in mast cells that are sensitized with the acylated preparation compared to those with the sialylated preparation. So there's definitely something inhibiting mast cell degranulation when we put on an acylated IgE on a mast cell. So we've been trying to really figure out what's going on. So we previously generated this panel of IgE antibodies where we mutated the glycosylation sites in each constant domain. These are all specific for ova, so there's identical antigen specificity. And we treated these IgE with buffer or with neuraminidase. So we have a panel of antigen-specific IgE with and without sialic acid with glycans at defined points in the molecule. And we tested this in the cutaneous anaphylaxis model. And so the sialylated, the buffer control treated animal IgEs triggered fairly robust anaphylaxis, as you can show here. And then when we essentially compared the neuraminidase treated to the buffer controls, what you can see is that it's only when we deplete the glycans in the CH1 domain do we lose the phenotype, this attenuation when you remove sialic acid. So we think that sialic acid removal affects glycans in this part of the molecule in the Fab, not anywhere else. And obviously we left out the mutants for the CH3 because that's where the oligomannose glycan is that's required for binding the high affinity receptor. So we thought those mutants would confound this response. So we made every iteration of double and single mutants of the glycans. And you can see here it's only the triple mutant that has this lost phenotype. So we think there's a sialic acid dosing effect that's required for this attenuation when you remove sialic acid. Right, so Shyant's in the lab has picked all this stuff up. And so he asked, what is the structure of the inhibitory glycoform? And so this is just an IgE molecule with these glycan schematics. And so we're not touching the oligomannose glycan. But he took IgE and treated with a series of glycosidases and then gave it to mice. And so some IgE was, so this is just the sialylated IgE shown here. He removed terminal sialic acid to have this galactosylated IgE. He then removed galactose to have IgE with these complex glycans that terminated in N-acetylglucosamine. And then he removed terminal N-acetylglucosamine sequentially. And then he treated IgE with endo-F2. That'll essentially will cleave off the most of the complex biotinary glycan, but it does not affect the oligomannose glycan. And so he put these different glycoengineered IgE antibodies into the PCA model, and these are the results. You can see that the sialylated IgE is pretty allergic. When you remove sialic acid, it's attenuated. It stays attenuated if you subsequently remove galactose or N-acetylglucosamine. But then when he clips off the entire glycan, it rebounds. So we think that essentially there is some inhibitory glycan that's either the mannose trimer or the bisecting N-acetylglucosamine that's being exposed when sialic acid is removed. And so we're trying to investigate specifically what that is and also trying to figure out what the receptor is that's interacting with this molecule. But This is globally. So we just did this on mouse IgE. And so, but so all of the sugars on IgE are being treated, but they need to be the complex bacterial structure to be affected by these enzymes. The oligomannose will not be affected. Yeah. Yeah. Exactly. Yeah. The endo F2 will basically cleaves here between these two N-acetylglucosamines, but it does not affect this one. So endo F1 is the one that's selected for the oligomannose, but I'll convince you of that. But yeah, so you can see that the endo F2 it's as strong anaphylaxis as the sialylated IgE, right? And so this is looking at essentially binding to allergens. You can see all of the different preparations bind to the allergen in like an oval ELISA, and then binding to the hyphenated receptor, you can see the octet traces, they all bind. And so this is essentially the endo F2 structure. So it still binds to the hyphenated receptor. So the oligomannose is intact, right? So so we don't know what's going on, but there's something about what type of glycans are present that makes IgE inhibitory. Yeah.
So like it's super cool, like all this stuff is awesome. And like you see this difference between the allergic and the non-allergic patients. So where is the the sialylation happening? Is it in the antibody stream cells, or is it in circulation?
No. So we presume it's happening as the antibodies are being made and then secreted. But it's certainly appealing to think about non-allergic IgE producing cells that might either not put on sialic acid or might remove it. But there's also neuraminases that are expressed at different places. Like there's surface neuraminases, there are lysosomal neuraminases. So one of the limitations for our studies, we only have access to the blood. So we can only get circulating IgE. And we can't resolve allergen-specific IgE. We had to do total IgE too. We just, by mass spec, we couldn't get enough to do any smaller population. So I would love to get tissue biopsies from Kari Naidu that has it down the GI tract and really look at what the IgE looks like there. But we can't resolve, sub.
You don't know if it's extracellular or negative regulation.
No idea. So, I mean, I joke about not being a cellular immunologist, and it's definitely true, but it's so complicated. And so we've just avoided that question altogether by engineering it, first, because it really is very difficult to answer, but we're super interested in it, for sure. Yeah.
Yeah, I know you're familiar with the effects of monoamine IgE and anaphylaxis, so we have a chance to look at, so we can moderate the oxygen for survival. So we had a chance to add these different forms of IgE on that, so without cross-linking the
Right. So that's the question is looking at what monomeric IgE does to mast cell survival, because IgE is a survival factor. We haven't done that, so we're really interested. We've been focusing on essentially at mast cell degranulation and not.
that may be even more important for the patient.
Sure. We really just haven't done it. I mean, part of it is like my attention span is only so long. We really want these short models, right? We want to get results so quickly. So, yeah.
But going back, you mentioned hypo-IgE syndrome. That, of course, is a much back. So there are some hypo-IgE syndromes that have a lot of allergies, and there are others who don't. And it depends on the defect, molecular defect. For instance, start 3, loss of function. They have lots of IgE, but not much of allergies. Whereas stock 8 deficiencies, which is autosomal recessive, they have lots of allergies, and it may be comparing the two populations of IgE proteins individually may show you a difference.
Sure. No, it's a good, like my dear friend Stephanie Eisenbarth is always in my ear about looking at Doc 8. But Jolon Walters passed through MGH for a while. And so she does have these cohorts of some of these patients that you're mentioning, this AT6 deficiency, Doc 8 deficiency, also Omen syndrome. And so we're starting to just really get into these samples, but just for lack of hands and turnover, we really haven't done it yet. But no, that's really an excellent point and something we're hoping to interrogate. It's all, everything we do is all passive. Yeah. So yeah. Hi. That's an excellent segue, actually. So if you'll humor me, and okay. So this is what we think is happening. But yeah, so allergic IgE, which we're calling pathogenic, has a ton of sialic acid, right? And so when you cross-link it, you get signaling and degranulation. When you remove sialic acid, we think there's some inhibitory signal that's being conveyed. And again, that's because when we remove the glycan here, it goes away. So we think we're trying to find out what the inhibitory glycan structure is and what the receptor is. So that's what we think. And yeah, because like the signaling is attenuated, anaphylaxis is attenuated, but again, it's just speculation, and I'm patching a bunch of conclusions together for sure. It's not totally clear, but we did an IP once actually with mast cells loaded with acyl IgE and actually mass spec DC sign and haven't been able to repeat it. So yeah, so there's part of me is hoping there will be DC sign and part of me hopes it won't be, but we have all the reagents if it is DC sign. So if I just have do I have time just for a couple more slides or? So we wanted to modulate sialic acid and IgE. So we basically made this Frankenstein molecule by putting a neuraminidase on an IgE-Fc instead of the Fabs, and it's still bound to the high-affinity receptor. So we could target neuraminidase to IgE-bearing cells. And this molecule was active. You can see it's just removing sialic acid. And then we put it on LED2 cells that were sensitized with sialylated IgE. And you can see when we titer on this neuraminidase IgE-Fc, we can actually start to attenuate degranulation of the mast cells in vitro. If there's essentially there's no allergen, you can see there's no non-specific degranulation. And then in vivo, we turned to that a model of systemic anaphylaxis where mice were sensitized systemically with the sialylated pathogenic IgE. They were given a neuraminidase Fc or an isotype control, and then the allergen, and we measured their temperature loss. And you can see there's a robust loss in temperature in the sensitized individuals or the individuals, the mice, or mice that were given the sialylated IgE and isotype control, but there's attenuation on the animals that were given the neuraminidase Fc. So we can actually attenuate anaphylaxis in vivo by modulating sialylation. All right, so yeah, so we went over the molecule, or the model rather, and so I hope I've convinced you that glycosylation is important. And so we've been spending a lot of time on sialic acid, but for IgE, sialic acid acts differently across the different subclasses. And then for IgE, there's this oligomannose that is essential for binding to the high-affinity receptor. And then sialic acid, we think here, this part of the molecule is important for setting the threshold for anaphylaxis. All right, and so I need to thank a lot of people. So these are all the former members of the lab that really made huge work, and the current lab members. So CIIT is currently our IgE guru, but formerly it was Kai and Michelle, and then Katrin, and then all of the work on the IgG story is being done by, who is it? So it's Anna and Sunny, and then funding and collaborators. So thank you so much. Sorry it ran a little bit over.